I guess what I plan to do today is, and Professor Datta suggested also, that we will spend maybe about an hour or so on to finish the course. So I will skip a lot of stuff, and then we will spend good amount of time, maybe half an hour to an hour, for discussing. And you may have questions you want to ask, you want to discuss anything. So we will just informally talk after that to conclude. So let's go ahead. We were talking about photothermal therapy. क्या है वो मैसेज स्लाइड इसको जरा डिलीट कर. So anyway, photothermal therapy, as I said, simply means that you use light. हो गया. So it just means that you use light to heat, and the heat is by the nanoparticle absorbing light and then transforming it, converting it to heat rather than emitting another photon. And for that, a number of materials have been used. You can also use dyes, organic dyes. As I said, if you use high concentration of dyes, then they don't emit. It's called concentration quenching. So the light absorbed is all converted into heat. But more effective is these metallic nanostructures like gold nanoparticles have been used extensively because they also strongly absorb and they don't emit. So all the light absorbed into plasma band is going to be converted into heat. So and so the idea is that you take these nanoparticles, again you target, it gets into the cells, and then you shine light, and the light will heat locally. The temperature you can even monitor, you can have 30 degree rise of local temperature, and that's high enough to kill the cell. Okay? But one of the things you want to do is that you want to again use the light which is in the wavelength where it penetrates deep inside the tissue. So you want to use in the window 1, which is around 1000 nanometer, window 2, which is around 12 something, window 3, which is around 1700 nanometers. So for that reason, if you use gold, say for example gold nanoparticles, you don't want to use a spherical solid nanoparticle because its plasma resonance is fixed around 530 nanometer in green, and that doesn't go deep. So there is a lot of activity using different kind of a structure, and we saw earlier that if you use core shell structure, with maybe core being like silica or core being some polymer, and you coat that with gold, then the resonance can be shifted to longer wavelength. Or if you use a rod, then in case of rod, surface plasma resonance along the length, longitudinal, is a longer wavelength, and it can be tuned further into longer wavelength by changing the size and the axial ratio. So there is a lot of interest going on. So this is like core shell structure that relaxes and converts to heat. Okay, but people have used this shell of metal or nano rods, or they have made nano cage, or they have made stars, and these ones again have plasma resonance that can be tuned to the IR. So there is a lot of activity in synthesizing these kinds of structures, of metallic nanostructures, and use them for thermal therapy. And again, is like treating cancer or getting in the cell and trying to heat it. This is gold embedded in polypeptide encapsulation, where you can do both chemo as well as photothermal therapy. So you can include the drugs in this micelle nanoparticle. You can also put a smaller gold nanoparticles, and you can do combined both. Okay. Another therapy that I want to talk about is gene therapy. Okay. And for that, you need to put a charge on the surface, and you need to put positive charge rather than negative charge, so that the DNA or RNA, they can actually bind electrostatically to the surface. So our original thing of nanocleaning, there was silica bubble. We coated it with something else, polyelectrolyte, some kind of polymer that has positive charge, and then you have positive charge, you can use it to bind. So let's talk about gene therapy. Gene therapy means you can deliver certain kind of genetic material to the cell, and it can do two kinds of functions depending on what kind of genetic material you put in there. One of them is called gene augmentation. So suppose your cells in the course of disease are lacking certain kind of genes, and that gene is not there, or that DNA is not there, and that's what causing the problem. So in that case, you identify the DNA that's underexpressed, that is not there enough to express it, to make proteins of the kind you need, then you have this disease, so you deliver that DNA. Okay? So in this case, it's called gene augmentation. So you have that nanoplex carrying DNA, it goes inside and gets into the endosomes inside the cell, which is where this gene is released, DNA, and the DNA can get into the nucleus and can replicate now and can replenish it. So if that happens, then the other one is called gene silencing. And gene silencing means that, some of the diseases like cancer may have oncogenes. This is the bad genes that are overexpressed. They're active, they're becoming more and more active. And so what you want to do in that case, you want to silence the genes, okay? You want to stop their overexpression. And that is done by delivering a small chain RNA. So siRNA. Okay, the siRNA is again also have negative charge. So you make the complementary siRNA. Remember, RNA is single-stranded. And so you bind it on nanoparticle, by electrostatically it gets delivered. And when it is released, this actually binds to your DNA, and then it stops. It binds irreversibly and stops it from functioning. Okay, and that's called silencing. So gene silencing. So when you silence oncogenes, then it's not, cancer cell is not proliferating, it's not replicating very fast because the process of replication has stopped. Okay? So these are the two techniques are used for very different diseases, and it's called gene therapy combined. And gene therapy is becoming more and more popular because that way you are manipulating the genes of the cells to treat the disease at the very fundamental level. So you can also use your imaging technique. So we showed how in nano formulation we can put optical probes on nanoparticles that carry the gene load, and so you can, by looking at the imaging again through this, you can see how the nanoparticle is moving to get to your target site, right? Carrying that genetic payload, okay? And so you can do binding, and you can study all these payload carrying over there. Then you can use light activation actually also to release it or monitor that the gene is being released and it's going to do the function over there. Then you can probe how it is getting into the nucleus, if it's DNA, and how the gene expression works. So all these can be dissected using this method. And so we are using gene therapy for a number of diseases, starting from Huntington disease, lung injury, chronic pain, obesity, asthma, stroke, infectious diseases, drug addiction were used. So in this chart you should read, it gives you like where we are adding gene to function and where we are silencing gene for certain disease, bad disease. So this is overall, and originally we use organically modified silica, like nanoclinic to carry that. Later work we use gold nanorods, where we can use the plasmonic property of gold to track it and also heat to release it. So this is the scenario of this. Here we showed organically modified silica with positive charge that binds to DNA. The positive charge is exposed on the surface, and then we also put an optical probe inside the nanoparticle, and in the DNA we intercalated another dye. Okay, and this dye, there are certain kind of dyes that get inside the double strand chain of the DNA and they stain the DNA. That's how you kind of monitor DNA also, by staining it with the DNA staining dye. Okay, so this dye is over here and is sitting on the nanoparticle, and when they're two together, these two dyes, you excite this dye. This is at a shorter wavelength. This emission is a longer wavelength. So there is energy transfer and emission is coming from here. But that happens only if the two are bound together. Okay? When this dissociates and you excite it, you will see emission from here. So by using this kind of nanoparticle formulation, we can track whether DNA is sitting on the nanoparticle and being transported, or whether it's released. Okay? It's saying that how it gets into the cells and we can monitor it. Here we again did imaging, and this is a case of drug addiction, which we worked by using gene silencing. So we showed that this dark 22, which is a biomarker for addiction, we can silence that by adding this siRNA in nanoformulation. So this silencing, this worked very well. This paper is also published. Then I want to talk to you a little bit about infectious disease. We have worked with flu influenza, flu, pneumonia, HIV, and tuberculosis. In this case, again, what we are doing is combining multiple drug delivery, because many of these infections, they become resistant to one drug, one antibiotic. Okay, so you want to actually use multiple drug delivery, and a very clear example is HIV. HIV, if you remember when it started, it was somebody got HIV. This was a kiss of death. That means that person is going to die eventually. But now nobody dies from HIV. The reason was at that time they're experimenting with single drugs. And for a long time they tried various drugs. It didn't work. And then when they entered the combination of drugs together, then now the person can take a drug every day, these multiple drugs, and can live a normal life. Okay, so that's multiple delivery, and then we can combine that with other kinds of therapy, photodynamic, that we saw, light activated therapy, or photothermal. Okay, or we can do this gene therapy that I talked to you about, where we can use gene silencing to silence, the infection that produces these bad genes. Or another one area is immunotherapy. That's a coming up therapy where you boost your own immune system to fight against the disease. So this is a very powerful tool, and there is a lot of interest, in trying to boost your immune system. Okay. So this is one of the nano formulation we made for tuberculosis, where we utilized this kind of configuration where we had a core was made of PLGA, polymeric nanoparticle, okay? And PLGA is hydrophobic mainly, okay? And so that trapped some of the chemotherapeutic agent used for tuberculosis. One of them is rifampicin, is very extensively used. So that's included. This is hydrophobic, is inside the hydrophobic region. Okay? And then we coated it with a layer of chitosan. This is a biopolymer, and it's made of saccharides, and this is hydrophilic. So we coat that over there, and then on the surface we connected this molecule called beta glucan. And beta glucan does immunotherapy. Okay, it intracellularly modulates this expression of your chemokines and cytokines, and generates a species to help treat the cancer, the infection. So it elicits immune response over there to fight against disease, and then when this nanoparticle degrades, both of these are biodegradable. Okay, when it degrades, the chemo is released, and that is chemotherapy. So we showed that both immunotherapy and the chemotherapy work together using this method. And this just shows the highlights of some of the things that our institute has produced, taking thick tumor, taking photodynamic therapy by upconversion, gene therapy, gene modulation, immunotherapy. So you can read that. And again, as I said, we work with a lot of different kind of formulations, but now we are only focused on two. If we are using inorganic, very rigid, they don't degrade, then we want to make them very, very small. If they are larger, polymeric nanoparticle, we want to make sure that they biodegrade. Example was that PLGA and chitosan that I showed to you earlier. They both degrade, fall apart. Another one is over here using fluoronic polymer encapsulation of, we will see later on, this is part of turmeric that you use, so natural drug, and we can use this that also degrades. Okay. So ultra small nanoparticles, we have used primarily core shell design, and one example is where we are looking at, they are 5 nanometer size. They can be used for imaging and therapy. And here the idea is that you can have a shell core, which could have sodium yttrium fluoride doped with either emitter or upconverter for photodynamic therapy to convert light, and there is another therapy that I'll tell you later about. And then you have a shell that contains gadolinium, and gadolinium can be used for MRI contrast. Okay? And we did a study of biocompatibility, as I said, by looking at how toxic they are to cells. It's called MTT assay. It's a standard assay to count live and dead. Remember I said the dead cells, they stain easily, but certain die, and use that to count. We also look at bioelimination that I explained to you how we do bioelimination studies. Then for brain, we wanted to use how we cross the blood-brain barrier first, and this is the kind of phantom we made. So you have kinds of cells at the bottom, then you have a membrane, then you have another kind of cell, and so then you can put these nanoparticles over there suspended. This is forming here, and you want to see how they migrate through this. Okay, yes?
Is it mandatory to follow any rule, like when we design a drug, so we have to follow the rule of this group 5. So as per the blood-brain barrier case, is it mandatory to follow any kind of rule to design a nanoparticle?
No, I'm not understanding your question. Repeat again.
In case of drug designing,
Drug delivery.
yeah, drug designing,
Designing,
Yeah.
yes.
we have to follow the rule of Philippines rule of 5. So the molecular weight should not be more than 500, as well as log P value and hydrogen bond donor as well as receptor. In the same case, while we design the nanoparticle, we have to follow any kind of rule that our nanoparticle should cross the blood-brain barriers. So we have to follow any kind of rule.
Well, first of all, you only need to cross blood-brain barrier if you're targeting your brain, central nervous system. If your cancer is someplace else, you don't need to worry about blood-brain barrier, right? And so if you want to cross the blood-brain barrier, there are certain kind of guidelines that, based on our experience, we feel that you should do in order for it to cross the blood-brain barrier. One of them is the size. And if the size is too big, it will not go through the blood-brain barrier. Second one is that there are certain kind of targeting group we can put on the surface, and those targeting group facilitates migration through this, okay? And and you can read one of our papers, we talk about it, and then charge on the surface also. So you manipulate with these things, then the best thing is to, once you follow these simple guidelines, then you actually experiment this in vitro model to see whether it crosses, okay? And then if you want to do in vivo, the real proof is in vivo. In vivo actually inject, and you can again through imaging or through dissection later on and looking at the brain and looking whether nanoparticles light up. If they have optical, you can see whether it has crossed. So modes crossing of the blood-brain barrier. By the way, so blood-brain barrier is sort of like tight junctions on cells. And this barrier, but not getting through it, but actually breaking it, rupturing it. And you want to make sure that it doesn't rupture the barrier, it just migrates through it. So the best way to monitor is that these barriers have electrical resistance. Okay, certain resistance. And if they are broken, the resistance will change. So by monitoring the electrical resistance, so just crossing across, and whether up to down, in this case is up to down, and seeing emission from down that doesn't migrate it through. At the same time, you should measure the resistance of the barrier and see if the resistance is changed quite a bit. If it is changed quite a bit, then that means it has forced its way and it's broken the barrier. Okay? Okay. Another kind of thing we did, and then we will move out of there, is that these gadolinium nanoparticles also can absorb X-ray. And when they absorb X-ray, they can release highly energetic electrons locally. and that highly energetic electrons can kill the cancer. This is what is called radiotherapy or radiation therapy, one model of that. Okay, so we are also using, so gadolinium does two things. First of all, it actually provides contrast for MRI, magnetic resonance imaging. It's magnetic. Second thing is that it enables you to use X-ray for therapy. Okay? As you know, many of the cancer you irradiate with X-ray or gamma ray or other things to kill it. That's called radiation therapy, right? So it enables that. Huh? Excuse me? Emission.
No, mutations.
Radiation. Mutation. Oh yeah, one. There is always a danger, and there is a dose dependent. So you can actually, the idea is that you use a dose at which the tumor cells interact first or kill and not the normal, because you want to prevent mutation in normal cells, not necessarily, you know. So it's a very established method, radiation therapy, but there is always a danger of mutation. Huh?
Cobalt.
Yeah, but that one is nuclear therapy. That's different. That is when you did actually radioactive thing inside. That's different than here. Here you're sending X-ray the same way as you do X-ray for X-ray image. Even you go to chest and others, you go to X-ray, you image it, or tooth, you image it. There is always danger of, you know, infection and others. But here, so you use externally incident X-ray, whereas the one you're talking about, cobalt and other, you actually it's radioactive. Huh? Yeah. Generating. And we'll talk about that later at the end, that therapy. So this is an example again of how you can think of formulations where the same component can do more than one thing. In this case, gadolinium is doing MRI and also doing enhancing radiation therapy. Okay. Then another thing, complexity you can add, and these are the particles, they're also called multifunctional. They do many things together. Okay. So another thing that we did, that we added patches of MnO2, manganese dioxide. Manganese dioxide, if you remember, this oxidation state of manganese is +4 over here, right? Fairly high. But manganese also forms Mn2 plus, divalent. Okay? So what happens is that you will see in a minute that manganese dioxide, let me just look at here. The manganese dioxide, when it gets into the tumor area where which is a little more acidic, it reacts with that, and there may be some peroxides or radicals in the, which is generally always called reactive oxygen species, ROS, that's also present in the cancer area. So it reacts together and it reduces to manganese 2 plus, releasing oxygen. And that oxygen can cure or remedy hypoxia, because many of if you have a big tumor, inside the tumor volume of cancer, there is regions where there is no oxygen, there is a lack of oxygen, hypoxia. Those regions are very difficult to treat either by chemo or by radiation therapy. So if this nanoparticle gets in there, you can image that area, you can release oxygen to take care of hypoxia, and then when you shine radiation, you can do radiation therapy more effectively. So this is the kind of example of how you can put together, build things to do multi-level, effects, and that's what we call nanoparticles are multifunctional. It does many things together. Okay. Now I'll tell a little bit about brain research. This is a very active area in America and many parts of the world. And here, if you use light to image or sense or activated therapy, I'll give you an example of that. The area is called neurophotonics. Neuro is for neurons in the brain, and using light wave. You can use chemistry to make nanocarriers. You can do imaging to map out the brain activity. You can use detect biomarkers for diagnosis of disease. Then you do therapies. Important part again is crossing the blood-brain barrier of the nanoparticle. Then also there is a lot of role for bioinformatics over there in computation, predictive modeling. So this is a very active field and multidisciplinary field. A lot of people are interested in that. We work with neurosurgeons, neuroscientists. So one of the things that we design nanoparticle, just to give an example of, is this excretable, again, small nanoparticle that can get out of the body by excretion. And this had multi-level. So you have a level in the core in the middle of sodium ytterbium fluoride containing thulium that takes a 980 nanometer light going deep and converts that into blue light, upconverts blue light. And this blue light does important thing, is absorbed by a genetic material called channel rhodopsin, and it stimulates the neurons. And that process is called optogenetics. So genetic material you have excited, and that actually makes neurons fire up, So you actually, this process is now considered very important tool to treat what is called cognitive disorders, loss of memory or impairment of memory or injury of brain, to concussion, so on. Optogenetics is a very exciting area. But the trouble again is that to excite it, you need a blue light. One common one, genetic material that is used to excite is called channel rhodopsin. It needs a blue light. And blue light doesn't go deep through your skull or brain. So that's why our nanoparticle can help. Here you only need an IR light to come here. And if this is sitting next to channel rhodopsin, it converts generates blue light that's absorbed by channel rhodopsin to give you optogenetics, okay? So that's one thing. Well, what does optogenetics then, how do you prove that the neurons are getting firing? One of them is that the ion channel activities of the cell, the ions, calcium, potassium, and all these ions, they go through ion channels and modulate, the potential and activities, okay? So one of the things was that if actually optogenetics is effective, then it open up the ion channel, then calcium ions influx can increase. So how can we probe that? We can probe that if you use a dye, right, that binds to calcium and changes its fluorescence. And these are called calcium dyes. You can buy them commercially, okay? Many of these dyes. So I want to have in an experiment that I want to do in the lab, in vitro, I want to excite channel rhodopsin, for which I need blue light. I want to then excite calcium dye to see whether there's more ions coming in there and the fluorescence is changing more with that, right? For that I need green light because the calcium probe, one of them is calcium green. This organic dye that binds to calcium and changes, one of them is calcium green, it needs green light. So I need over the green light, okay? And then if I want to image optically, I need near infrared light, 800 or 1000 nanometer light that can go deep, and we have emission coming out. So how do we generate all these lights? I need 3 different colors. Okay, so this is where we have this design. This one, they all take 980 nanometers. This converts to blue that I need for optogenetics. Then there is a shell containing erbium ion. And erbium generates green that I can use to probe the calcium ion activity, excite the calcium dye. And then the outer layer, not in this configuration, but the others, I can put again another emitter, okay, that emits in the near infrared range to do optical imaging. Okay, so I can combine all these 3 functions in one nanoparticle by using core shell structure. Okay? In this case, happens that it has gadolinium outside only, and gadolinium is for MRI again. So rather than optical imaging, we could use MRI. Okay? Again, you read about it, neurophotonics, what is the scope is. So here, one of them is imaging. We do, and again, we showed that actually if you use a light in the near infrared at 980 nanometers, and your emission is at 800 nanometers, that conversion that we talked about earlier, you can actually image through the skull. You don't need to get light by making a hole in the skull and fiber. You can directly just send the light at 980 and collect it and image it. So that was one thing. But again, the image you see is a little bit blurred. This was the pattern that we wanted to image. It was a little bit blurred, and you need a lot of image processing. This is where, modeling simulation comes in, how you can correct and do different kind of image processing to enhance the contrast. Okay? This is the concept. Basically, I already told you about the bacterioresorption, which is here in the ion junctions, and in the membranes, and you excite it with blue light, and it opens up the ion channels for calcium ion to get in there, firing of neurons. Another one we want to talk about, give an example of brain tumor therapy, so brain cancer. One of the more frequent and deadly cancer of brain is glioblastoma, okay? And it's a frequent aggressive brain tumor and very low survival rate. And surgery in the brain to remove it or doing chemotherapy have not been that effective. So one of the things we are now looking at, and this is where I was thinking that India can also do a lot, is natural medicine. How we can make nano formulations of natural medicine and use them for treatment. So here we are talking about turmeric, and the important ingredient that is curcumin. This is the structure of curcumin. And people have postulated that it actually inhibits the tumor growth. Okay, so but it is actually, turmeric, as you know, is hydrophobic mainly. If you put it in water and try to stir it, it's not going to dissolve fully. Maybe a little bit, it doesn't dissolve. Okay, so it kind of aggregates around, and so one of the things is that how we can use is to entrap, and then can be delivered to the tumor site by targeting. And the other one also is that curcumin has optical activity. It's a yellow color, so it absorbs in certain regions, mainly in the green, and then it emits light too. There is a weak emission from turmeric also. So you have emission. So it's a photonic, it has a photonic signal. So we can track it if it's a nanoparticle. By looking at the emission of turmeric, we can track where it's going. So here is what we did. And then we use this pluronic polymer. This is the structure of that, where it has some positive charge. And so we've made a nanoparticle that has curcumin trapped inside this pluronic polymer, which has the tails coming out, the and these tails at the end have a charge. Okay, and then we encapsulated another dye that absorbs at the same wavelength where turmeric emits, so that there is energy transfer from turmeric to this dye. And it emits, this dye emits in the near infrared. So we can monitor by energy transfer, the same thing we showed to you in gene therapy. We can monitor whether this turmeric and this molecule are sitting together. If we excite turmeric and it emits light from here, we can monitor. So we monitor that it crosses blood-brain barrier, these nanoparticles. Then we have targeting groups around here. We deliver it to the site. When it goes to the site, and this is biodegradable, when it degrades, this dye and this are separated. Then when you excite curcumin, there is no emission coming out from this dye. So we say this is delivered. So we can make this kind of package, and we did it. We did also in vitro, in vivo studies in mice, and indeed it looks like it, these are the details, it looks like that the cancer actually is shrinking the tumor with this. So we did in the animal study itself, And then another one we have been doing using ginseng. In India, you also have something similar to ginseng. What is it called? I think it's called ashwagandha. Yeah, that's similar to ginseng, right? Some people call Indian ginseng also. So, and ginseng people have also said that helps in Alzheimer. So we have been experimenting now. This is also hydrophobic. So we encapsulate in PLGA, chitosan, and then have targeting group. We tested blood-brain barrier crossing, and then we actually have looked at one of the signature is this beta amyloid plaques are formed when there is Alzheimer, and we have kind of seen how we can label that using nanoparticle emitter, and how when release of the ginseng, it breaks up these plaques, okay? So that's another experiment we're going on. Now, then I come to another area that I'm very excited about. So nuclear nanomedicine, okay? So what is nuclear medicine? One part of nuclear medicine is imaging, which is called radiography, and it uses the radioactive isotopes, okay, and that's what provides the signal. So the radioactive isotope, gets into the site where you want to go, and then you can image by detecting the radiation coming out from radioactive nucleus, okay. So you have a SPECT imaging. PET imaging is a very powerful imaging tool, now very extensively utilized, and that uses fluorine-18, copper-64, iodine-124. So the idea is, how can we embed this in a nano formulation? And then we have targeting group to go to the right side, and then we can image more effectively, because the radioactive nucleus is mainly now at the tumor, not at healthy tissues, and we can map out. So that's kind of thing again. Here there is a challenge for chemists, because these ones, you remember, radioactive nucleus has certain half-life, and during that period its concentration drops to half, right? And so you need to make sure that your nanoparticle is made with this in that time, deliver, and it finds its way to where it is supposed to go, and you can image, okay? So for that, you may want to use a radioactive nucleus that has a longer lifetime, right? On the other hand, you don't want a radioactive nucleus to have too long of a lifetime, because, after the tumor is gone, it still is radioactive. And if it's still radioactive, it can hurt the other areas, normal tissues. So you want to have a balance where it is not so long, but it's still not so short, that it has time to do chemistry. So how chemistry can help? If you want to have 4-step synthesis or encapsulation using radioactive nucleus, it's not practical. And it's not practical to make in an ordinary lab, because you don't want to use a radioactive atom or compound. You need to have special protective labs, wearing gloves, thick gloves, cover shield, and also have a glass made maybe containing lead that absorbs these, okay? So there are precautions to be taken. And then you need to have a chemistry that is very fast, okay? So there are a number of chemistries people use. One of them, what we call self-assembling, which is not a step synthesis where you heat and conduct reaction for a while. You mix things together, they very rapidly form something, okay? And they can trap radioactive compound in there. And so one important example could be these metal organic frameworks, right? That can very rapidly form, and enclose this. The other one could be where you have formed a nanoparticle, but it has some anchoring group on the surface, carboxyl group, amine groups, or thiol groups, or whatever it takes. And your radioactive compound has the group to immediately attach on the surface. Okay? And if you're talking about radioactive metals, you may want to have a group on the surface that forms as a ligand. Right? And when you put these metal ions, radioactive, the ligand immediately attaches to it. So you want to think about this clever chemistry and design, and that's another opportunity for chemists, how we can make radioactive nanoparticles that are, require very short time of coupling, okay, or incorporation. All right, so the other one, so what is the advantage of nanotechnology over here? The concentration of nuclear agents at the target site could be high, because they all go over there because of targeting. The second one is, so related to that, is that then the damage to the healthy tissues is minimized, is all going targeted to there. Okay. The other one is we call nuclear medicine. So by the way, this is kind of either called nuclear nanomedicine or nanoradiopharmaceuticals. It's like a radiation pharmacy, okay, treatment. Okay. So the other part is nuclear medicine, means nuclear therapy. And nuclear therapy can be of 3 different kinds. One of them is what we call photon radiation therapy. So you have a radioactive source that generates X-ray or gamma ray. These are highly energetic photons, okay? They are absorbed by the tissue themselves, or like gadolinium I showed to you in nanoparticle, to eject electrons and do therapy. So that one is what is called photon radiation therapy. Very highly energetic photons, X-ray, gamma ray doing the action. The other one is particle radiation therapy, where we bombard the tumor with a certain kind of particle, like neutrons, protons, alpha ray, beta ray, carbon ions. So these ones are particles. They're not photons, right? And they go and hit, and with their energies, they damage the tumor. Okay? So one of them is a very popular one is a neutron therapy, and we'll talk about that. Another one which is very, very also exciting, and many cancer centers now are having, is proton therapy. So you bombard that, so you have an accelerator that accelerates and gets the proton of right energy coming out, and then you focus the proton on the tumor, and then the tumor is destroyed. Okay, and so that's one therapy. The other one is neutron, and the neutron again is bombarded, so you have a neutron source. But more popular one is neutron capture, which you will see in a minute what that is. Then the third one is using radionucleotide. So this is actually incorporating again radioactive nucleus that is on site emitting beta ray, alpha ray, some of these high energy radiation, that can damage the cell. Okay? So these are internal source where they are generating these particles. Okay? So this is an active area and just starting to emerge how we can take advantage of. It has been active, all of them, without nanoformulation. Now the active area is how we can use nanoformulation to enhance it. Okay? So one of them is, let's say, neutron therapy is neutron capture therapy. And the idea over here is the boron. In natural boron atom has two isotopes, boron 10 and boron 11. And you know what isotope abundance is. Percentage of each of these isotopes in natural sample. So in natural sample of boron, you take boron compound, boron hydride and others, is 20% is boron 10 and 80% is boron 11, or maybe a small amount other isotopes. So boron 10, when you bombard neutron beam, it captures a neutron and it goes to boron 11 in excited state, and that boron 11 then decays, breaks down, it's unstable, it breaks down into helium particle and lithium particle. And both of them can do a lot of damage, but primarily it's helium. Okay? It does the damage. So see, this is called boron capture therapy. And it's not something, concept is not new. This concept was originated 15 years ago. And people were very excited. They used different boron compound. You can borohydrides and all kinds of other compounds, boranes, or borates, but all these compounds, they are molecular compounds, they disperse. Their concentration at the tumor site was not high enough to make it very effective. So now what we are doing, we are making nanoparticles that are totally made of boron. And how we do it, very simple. Like we did in case of silicon nanoparticle, we take a hydrosilane and we use laser to pyrolyze it and make silicon. Similarly, we can say hydroborane gas, and we pyrolyze it. All the boron atoms come together to make a boron nanoparticle. So we have made boron nanoparticles, and there is a little surface coating of boron oxide, because it oxidizes a little bit. But that still has a very heavy concentration of boron in there. And we are now experimenting that for the boron, this neutron capture therapy, showing a lot of promise. A lot of people are excited now because of that. Huh? We are using right now natural, but this is a very good point. Any of these, as you know, if you pay price, you can have enriched sample. That means you can get a sample which is maybe 99% boron 10 only, no boron 11. So that will be much more effective. That also is very expensive. So we are experimenting only with natural boron, which means has 20%. Huh? 20%. Boron 11 is 80%. Okay. So we have, so far what we have done in boron is that we have nanoparticles, we have conjugated it, we're targeting, it's a small molecule, folic acid does the targeting, and then we have dyes we have attached for imaging, and we have looked at cellular uptake, how the nanoparticles get inside the cell. And so we have done a lot of characterization. Their sizes range from 18 nanometer to 70 nanometer. So we can select which size could be most effective for generating alpha particle. We have done their toxicity studies, and they show that fairly they are non-toxic. Cell viability is very good. Then now I want to say a few slides about what our dreams are, and one of them is working at this health and wellness through lifespan. Okay, so we want to treat neurocognitive disorder from very beginning, embryonic state where we can see the signature, whether the child born is going to be autistic or retarded, and if we do that, how are we going to treat it? We can use optogenetics, gene therapy, chemotherapy, and all these in embryo itself. Okay. Then in the youth, they're playing football, or they're doing whatever other sports, or they get hit in their head, something, they have concussions, they may have brain injury. This is a very active area, it's called TBI, traumatic brain injury. Okay. So how do we correct the damage to the brain cells and repair it? So that's another one. And then the third one is, of course, elderly population, you have Alzheimer to deal with. Okay, so how we can treat this? So first step is assessing the brain function. And in our thing is quantifying the brain function. Okay, so that we can do it by functional imaging, different imaging probes, look at the cells, their activity, their metabolism, and all those to gauge that. Then once we have it, then we assess what is the extent of damage, which part of the brain is damaged, what we need to be replenished or connected, and one powerful tool we want to do is, as I said, one imaging we use is photoacoustic imaging, also combined. So MRI, photoacoustic, optical imaging, all these. Okay. Another important thing is gene therapy and this optogenetics that he talked about. There's a selected brain region to stimulate the neurons, fire them, okay, by using optogenetic. So our vision is identify and treat cognitive deficit from very beginning, okay. The other one is this, longevity, right? Everybody wants to, I don't want to live for a long, long time, but a dream is, can we live forever? Or in this case, let's limit to 1,000 years. And this has been, so imagination has been okay. You can find a fountain of youth, but you can find some miracle drug, you take it, and you can live forever. Or you have blessing of goddess, right? And that can grant you, right, immortality. But is it real to expect it? And the answer is yes. Okay? And so it's yes, it is possible, and there are very clear pathways for longevity. Okay? And so my one chapter in my book really talks about what I call rejuvenation therapy. Rejuvenation therapy. Okay? And the idea, this is the book, the idea is that what makes us age and die? There are a number of factors. Okay? One factor is, then in the course of time we connect a lot of toxins, heavy metals, reactive oxygen species, they're all growing in there in our body, right? And so we can use nanoparticles as a sponge to remove these. It's chemistry. Very simple, if you want to start heavy metal, you can do chelation chemistry. So nanoparticles can have chelating group, they act like a sponge, they go through this and start sucking up, right? Getting in there. The other one is you want to do neurological repair. That for there you can use gene therapy or a stem cell therapy, to replenish it. Then you have growth hormones to replenish the cells and make them healthy. Tissue engineering, so that's a very active area. Now even talking about organ printing. So in the laboratory you can order a kidney and it prints and replace your old kidney and put your new kidney. Okay, so it's like a car, you can rebuild the whole thing, right? And you can go for another 100 years, then you rebuild again, and you can go for 1,000 years. Okay? So longevity is not just imagination now, it's not a fantasy imagination. We can see that in my lifetime, I see life expectancy has increased tremendously. In America, somebody who is 70 years old does not consider himself old. He's thinking, well, I can definitely easily, I think I can live up to 100 years. Okay? In India, people used to die at 45 or 50. I remember when I was growing up, somebody who's 45 considers himself or herself old. I don't think you consider 45-year-old person considers himself old here. So things are increasing. Okay, so where are we at? Let's conclude and then we sit down and talk a little bit. Where are we at nanomedicine? In vitro diagnosis, there are a number of nano tools available already. And one example, I gave you colorimetric example, a pregnancy test, where you have a strip, and a strip of nanoparticles of gold that has a surface containing antibody. And then when we put urine on the top, and it has signature of pregnancy, certain antigens that bind on the surface, stay there, so and then it changes the color of plasma resonance of the metal film, metal nanoparticles. So the color changes, and by that you can detect, right? That's the first step. So that exists. There are several nano formulations of drugs that are FDA approved, Food and Drug approved. Okay, these drugs are already being used, but their main function is bioavailability. These are hydrophobic drugs. There are certain nano formulations that more easily enhance the delivery. Okay, so that one is there. A nanoparticle-based photothermal therapy is in clinical trial. That told you gold nano stars, gold nano rods, all those people are already doing experiments where they can be lodged into tumor and use light, infrared light, near infrared light to heat and kill. X-ray nanotherapy. So this is the company that we started in France, Nanobiotics. That's also now finishing clinical trial where they use nanoparticles that absorb X-ray, as I showed you, like gadolinium, and generate reactive oxygen species around through electron, and that kills. So this is already also in a year or two will be in patient care trial. So there are some examples of already in progress. Now, a lot of people who are critical of nanomedicine may say, well, look, nanomedicine started 15 years ago. So why aren't we seeing more nanomedicine? Why is it that you guys only talk about nanomedicine doing this, nanomedicine very effective, nanomedicine being safe? Where is it? Okay, and again, idea, as I said, that in the beginning, in any area, there is a lot of excitement and a lot of promises made. But real development of technology, especially in healthcare, takes a long time. So many people started different nano formulations, and 80% of them went nowhere. Okay? And now people are focusing more and more on what are the real nano formulation needed. And so there are many clinical trials already going on. and many new ideas like optogenetics and all those are coming into picture. So what can we look for? We can look for this more analysis and diagnosis, laboratory based. That's the first step, that's easiest to do. What I call liquid biopsy. You take saliva, you can take blood, you can take urine, you can take stool suspensions, extracts, and you can use those and diagnose. Look at the biomarker signatures. A nanoparticle allow you to look for many biomarkers, not just one signature, many, right? Another area is very active is breath condensate. So you just breathe on a strip and it captures some of the molecules expressed there, and based on that you do diagnosis. Implantable in vivo sensors that can be externally powered, not have things coming out, and their nano sensors can play an important role because you need to miniaturize the sizes and response. Gene therapy, immunotherapy, as I said, is a very important area. A stem cell biotechnology, I didn't get into stem cells, but there is a lot of advantage. A nanoparticle technology, nanomedicine approach also helps in preparing stem cells. So just 1 minute about stem cells. You know that there are two kinds of stem cells. One is the stem cells that come from, you can say like virgin stem cell, it's totally blank cells, right? And the other one, embryonic stem cells, okay, that you derive from embryo. And the other one is what's called adult stem cell. They are slightly specialized already, and they are easier to get, but they are not general that you can use and make it into different kinds of cells. So that was a big bottleneck. And then people discovered a way that you take these adult stem cells that already have certain genes acting, specialized, and they say, okay, let's silence those. Then it's like a blank. That's like embryonic stem cell. Okay, and those are called induced pluripotent cells, IPC, okay, cells. And then we rewrite on them, insert genes to specialize them. So that opened up a new doors of stem cells using. A nanoparticle again can deliver certain kind of siRNA to silence, and then another gene to make it specialized. So again, nanoparticle gene therapy has a big advantage combining that. Now we will even talking about you don't need to silence and all. You can do transdifferentiation. Then in one step you can go from one kind of cell to another. Okay. Tissue engineering is very active where nanoparticles can also provide a template to grow cells on that. And the rejuvenation we talked about is something to look forward to also. A new generation of carriers, you don't need to synthesize many things. There is a big emphasis on using biomaterials or bio-derived materials or agro-derived materials, natural plants and others. Like one of you were talking about a natural plant extract, and you can make nanoparticles from that, as well as you can use the drugs from the natural medicine, green materials. So green chemistry is gaining where we can actually use synthesis in water, not in organic solvents, which are toxic. One of the things we have experimented is in biosynthesis. So you can actually, it's kind of interesting thing that bacteria all the time make things, they make proteins, they make other kinds of molecules. So what about if we take the cells of bacteria and inside the cell, we can use their enzyme to make reactions. Okay, that's called biosynthesis. So one of the things we did, which was kind of very interesting, as I said, heavy metals are toxic, lead, cadmium and all these, right? But there are certain they're toxic to human, they're toxic to bacteria, okay? But there are certain kind of bacteria and These are called extremophiles. They're very robust and they survive in any kind of thing. So where do you find them? One example, some of the extremophiles is in Antarctica. The temperature is way, way down, and then it can come up to way up, extreme temperatures, and these bacteria survive. They're called extremophiles. So we took these extremophiles and their cells, we injected cadmium salt, cadmium source. We injected source of sulfide or selenide, like sodium, and we get in there and they synthesize cadmium sulfide or cadmium selenide, nanoparticles. They connect them, and then in course of time it grows and forms nanoparticles. So we made quantum dots using this biosynthesis, and the bacteria are still alive. You can put lead and you can make lead sulfide, lead selenide, and they survive. Okay? And so that's making biosynthesis. Well, the other side is, titanium metal is very strong and very resistant. That's why I use titanium implants and all these things. So people make now ships and other things that are to be protected with titanium metal shield right around that. So one of the areas is that how can we actually design things that even those are not resistant, they can be eaten up. So you use these extremophiles that have appetite for titanium, they can go and dissolve them and break their structure. So that's like, okay, so challenge is the future directions. I summarize few of these: multimodal imaging, spectroscopy, multiplex diagnostic, real-time migrating. We need to understand how the drugs interact with cells, what compartment. So now that's the area which is personalized medicine. They look at and they say, we cannot write generic prescription for every person for same disease, even the disease is the same. So how do we do that? We need to look at these interactions at molecular level. Distribution, clearing, crossing biological barrier, these studies, these are all for you to look at, choose one of these areas to do research and write thesis. Targeted delivery, control release, stem cell, and all combining therapies together by using clever nanoformulation. Okay, making new materials and opportunities, using a spectroscopy to quantify and look at different kind of diagnostic approaches, imaging, using longer wavelength light, again released by image-guided therapy is an important area, nanoradiopharmaceuticals, tumor oxygenation, neurotechnology, so a whole range of things. And then I listed some of the people whose work I have quoted here. So thank you for so the course formally is finished. Now I sit down and you ask and we can discuss in general. You can say what you are interested in doing. I can give you some suggestions, or if you have questions we can have. So let's spend at least half an hour discussing it. Okay? So let's go back to these and these slides, and you can particularly this one is a long list. All right. Yeah. Yes, yes. Yeah. You put in sweets. You put them there on the top.
So I think it is because of the nano sheet, or
well, silver has antimicrobial activity, and people in India realized long time ago that silver thin films which break down into little pieces and then they provide large surface area to interact, and it produces silver ion which is actually does antimicrobial activity. So they are not really nanoparticles. The film is not nano, but it breaks into little pieces, micro pieces, so they are still active. But now the new is that maybe nano formulation of silver will be even better. Nano silver. So a lot of experiments are being done by using nanoparticles of silver, because the nano has the largest surface to volume ratio. Okay. So there is an interest in that. Yeah. But for food, you don't want, it'll be too expensive, because making nano silver is not as inexpensive as a thin film of silver that you normally use for mithai ke liye without that, So.
What about homeopathy?
Huh?
Yeah, homeopathy. Yeah.
Homeopathy is an interesting therapeutic approach. In the US, they don't believe in it. So there are very few places where you would find homeopathic medicine. Okay? You would not find most places homeopathic treatment. India probably is the most used homeopathic medicine. But surprisingly, in Europe also they believe in homeopathy. So if you go to a pharmacy, I was in France recently, and I had to go to pharmacy for some medicine, mainly skin thing, and they offered homeopathic medicine. They said, we also have homeopathic medicine that you take, and so the reason is that now you have a homeopathic college and they teach things, but before, when it started, it was like Ayurvedic medicine. It was on belief that this cures, like haldi cures this, and neem cures this, and you believe in this and you practice. And now there is more scientific studies being done, and fortunately the results are coming out positive, that indeed this natural medicine that India believed for long time, generations, may have some scientific validity. Okay? So what I'm trying to say is I have not seen a lot of systematic scientific studies of homeopathic medicine. Okay? So it's difficult to say, but in America we call it placebo effect. Placebo effect is that if you believe blindly in something, whether it has any basis or not, it will work on you. So you take a pill and you say, oh, this is going to help me, cure me, and you take a pill, sure enough it will help you. Right? And that's called placebo effect. People have experimented it. They give blank something that was not a medicine, and they said, go and take this thing, and you feel better. And the guy goes and takes this thing. After 3 days, he says, yeah, doc, I feel much better. Right? So it's mind over matter, right? It's just mind saying, yes, I'm going to feel better with this medicine. So I think, maybe it is not really real, but the only thing can distinguish it by systematic study. और चलो कुछ और क्वेश्चन, discussions, anything?
Then my part of question.
Yeah, the students should do. You're here sitting there. Let's talk.
So how economic is this approach of using nanoparticles for healthcare?
Well, it's a very good question. I think that eventually, no matter how wonderful, magical this nanomedicine approach is, but if it is not affordable, most people are not going to use it. So, like any drug, if a pharmaceutical company develops a drug, it is a long process and very expensive, because they don't make one drug. They make one chemical structure, and they vary this to combinatorial chemistry. They make all kinds of things. They study their toxicity, then they study their drug uptake, then they look at the therapeutic value, pharmacodynamics and all those. So long process, and then they go through clinical trials and all these. So they have invested by then hundreds of millions, maybe $1 billion, right? So the drug, when it comes out in the market and they have a patent, is very expensive. Any new drug is very expensive. And then when it opens up and becomes generic, price drops, right? So I don't think nanomedicine in the beginning will be cheap. And it may be affordable only by certain population. I don't know if insurance will cover in the beginning. They always try to kind of get out of their commitment and say, oh, this is not fully established, I don't want to cover this, even if it is approved by FDA. Okay, so I think you have to pay from the pocket, then people can only certain population can afford. But how can you make it affordable? This is where you need to have new processing methods that you can make in large bulk quality. You can, if it involves many synthesis, you want to find an alternate route that it actually doesn't involve so many steps. So materials preparation and loading of drugs and batch processing. Maybe you can make robotics to make it. For India, it may not make sense. We have so many people that need a job. You don't need to push robotics here, right? So I think the idea is that you can definitely use and help in lowering the cost, So eventually it has to be affordable, otherwise it's a useless exercise.
Which one is specific ways that one has to use it?
Well, you're right. Look, what happens is the people who has a terminal illness or they have given up on conventional medicine or anything, then they are ready to try anything. I get emails from people who read my paper and say, look, my uncle or my father or my so-and-so has this thing and there is no cure for that. Can we try your method? And we are ready to sign anything and we are ready to try it on us, okay? And I wish I could help them, but I can't do it. Unless I have FDA approval, I cannot give any medicine, any injection to anybody, right? So I had to write to them that, look, it's a very early experimental stage. We don't have permission to try it on you, so we wish we could help, but we can't. Yeah. Pardon me?
So on what basis, but in the lasers there are two, pulse wave and continuous wave. On what basis we will choose these?
Okay.
The pulse wave we have to use or continuous?
Good question. Firstly, the answer to your question, we need to use a laser, is not true. Yeah.
Not that, but the particular wavelength of light.
Yes.
Yes, sir.
Yeah. But you have a broad distribution. There are also particular wavelength of light. You can buy solid state lasers which are almost monochromatic, or you can use light emitting diode. Okay. So what area is, can we give lasers? Laser is a pinpoint. So suppose you have a very tiny tumor and you want only expose that to light around that. The laser is the most suited. A lamp and LED are divergent, right? And that's one thing. The second thing is that if you want to do one photon therapy, so one photon is absorbed and it emits and does the therapy, then in that case, you don't need to use a pulse laser, expensive laser. CW lasers, continuous wave lasers, light source is enough. Okay? And so currently most of the photodynamic therapy are done with a continuous wave laser, not pulse laser. You don't need to do that. But if you are doing up conversion, like you convert the light to lower energy going up, and then do the therapy, if you do the nanoparticle with a rare earth, you can still use continuous wave laser or low power laser. But if you do organic dye by the virtual state 2 photon absorption, then you need a pulse laser. Then you need picosecond or femtosecond lasers, which are expensive. Okay? Okay. Or
Sir, why gadolinium is preferably used for MRI?
no, it is not preferably used for MRI. Well, the reason gadolinium is used because it has a high relaxivity, it changes the relaxation time quite a bit, significantly. Okay, there are 3 elements that are used for MRI. One is iron, so in form of iron oxide, and iron oxide nanoparticles have been extensively used. They were proven to be biocompatible, but now there are some concerns about iron oxide nanoparticles. The second one is gadolinium. The gadolinium has not been used in nanoformulation. They have been used in chelating form. So with a chelate. So this is a solution with gadolinium ion with a chelating group, and they use that for contrast. People now have said that gadolinium causes toxicity, gadolinium ion. So there is a problem with that, using this chelate compound. Okay, so our nano formulation where we put gadolinium in a fluoride lattice, gadolinium fluoride bond is not ionic fully. It's mostly covalent, so it doesn't break that easily. So it remains tightly held in the lattice. And if the nanoparticles are eliminated from the body because they're small, then gadolinium goes out. Okay, it doesn't stay in the body. So that's the advantage of that. That's why we are excited about gadolinium. The third one is manganese, Mn2+. That also have unpaired spin. You can use any ions or atoms that has unpaired spin for MRI. Okay, so manganese has been used, but manganese is not as effective. It doesn't have enough spin interaction to change a lot, so it is not as effective as iron oxide and gadolinium. But there is a lot of interest in manganese, using manganese as a contrast too. So you can think of a compound which is rich in manganese ion, a nanoformulation, which is rich in manganese ion, and then you have, or divalent manganese, that will work as a contrast agent, huh?
D5 system.
Yeah.
So in nanocarrier formulation, we are using lanthanides.
Lanthanides, yes. Lanthanides in what form? Fluoride, oxide, what? Which one?
So, I'm asking that can we use actinides also?
Can you do what?
Actinides also?
Yes, you can use actinides, but it depends on what you want to achieve. Lanthanide, you just don't use lanthanide. You use for it a specific property like gadolinium. We use for MRI. Gadolinium we choose also for this radiotherapy because it absorbs X-ray and generates thing. So and if you use like erbium, I use erbium for emission. It takes the IR and converts that in different wavelengths, right? Or thulium. So you use the appropriate lanthanides for this property. So similarly in actinide, you need to choose which actinide you want to use and why you want to use it, what property of the actinide you want to use it.
So these actinoids are radioactive nature, they may harm the other cells.
Hmm?
So as we know that radioactives are actinoids are radioactive in nature. They will affect the other cells or will harm the other cells if we will use these actinoids in beauties.
So you're saying that if you use radioactive actinides and they will harm, well, the idea is this, that you use a nano formulation with targeting,
Lengthenoids
you
are
know.
better.
Huh?
So lengthenoids are better. So lengthenoids are better.
Again, not all actinide elements are radioactive, you know. So you need to see what you want to accomplish. It's not one element being better than other. You need to look at what element, what property of that element you want to use. You cannot use erbium for generating light in the blue and UV. You need to use thulium ion because it doesn't have energy levels. So you need to select on the basis of the properties of these elements or atoms to choose. When you're talking about radioactivity and you have nanoparticles radioactive, you want to put right into the tumor, these nanoparticles so that they're not in a healthy area and they're exposed to radioactivity. Okay?
Why do we use nanoparticles, not smaller or bigger particles like what is technology?
Why do we use what?
Nanoparticles for these technologies. Why don't we use smaller or bigger particles in that?
What do you mean by smaller or bigger?
Microparticles or
larger range than
Larger micron-sized particles are not good for intracellular therapy because your cell dimension itself is 10 to 20 micron. How are you going to push micron-sized nanoparticle inside there? If you push it in there, it will break inside the cellular structure, like mitochondria, nucleus, and any of these will be damaged. Okay? So if you want to do, and the other thing is that through blood capillaries, the nanoparticles can move much more readily than micron-sized bigger one. They may attach to the walls of the capillary, eventually clog up. So nano is a small size and it provides to you, okay, also opportunities to encapsulate smaller molecules either inside or conjugate on the surface. It has a large surface area. Yeah.
Nowadays, it is proven that the lipids nanoparticles, that is SLN or NLC, are better than the polymeric nanoparticles because they are having low toxicity as well as they are easy to formulate, they are bioavailable and all these things. But so the problem is that while formulating the lipid nanoparticles, the lipids used are very expensive. So, can you suggest some lipids that is economical?
Well, first of all, there are certain advantages of lipid nanoparticles, okay? But there are also certain disadvantages. The advantage is that, as you said, they're easy to form, and they are also, in many cases, safer. The disadvantage is that they are not rigid enough, and unless you use solid lipid nanoparticle, that's another kind of lipids, they are like not stable enough generally for many applications. So it's difficult for me to give you general advice. You need to think of a project, and I know some of you are only working at making nanoparticles and characterizing and seeing what forms, what properties it might have. But if you think beyond that, well, where will you use it? Or what are you targeting? What would you want to deliver? Is it for a certain cancer, a certain chemo drug? Then the topical lipids are better because they they're like oil, they form a very nice emulsion and they can be absorbed transdermally, okay, in there. So in terms of cost, I can't give you advice. I don't know what is available here and what the costs are, what will be cheaper, but I'm sure there are a whole library of lipids, and some of them you can derive from maybe natural source, and make it a natural source lipids will be much cheaper. you know. So it's for me, on the cost-wise advice. और हाँ बोलिए. Can you repeat the question again?
Nanoparticles.
Well, I think that natural medicine definitely has merit. But in case of natural medicine, you don't want to start something that has not really proven to have some basis for its activity. You know, this thing cures cancer, and you start going and making nanoformulation of that. Before that, you need to have some reason to believe. There are some studies that are saying, okay, we have seen anti-cancer activity of this. But turmeric there are some studies where they have already shown some activities. That's why we took it and encapsulated it to see whether we can make it more effective. Because turmeric is hydrophobic and it doesn't get well inside. But otherwise, if somebody says, well, I think this is a very good, I wouldn't jump and make a nanoformulation. Firstly, I want to see, does it really have anti-cancer activity? I cannot hear you. You speak softly and there are people talking. Go ahead.
Sir, I'm saying biochemistry student, I can analyze the effect of all these natural products and
then encapsulate it, yes. So first one will be that you can take the drug and you can mix it with surfactant and simply disperse it and do cell toxicity studies. And if that shows that there is actually cell killing and so on, then you want to make a nanoformulation and make it more effective. Yes. बोलिए.
Like this,
Yeah.
let have our snacks and tea first.
Yeah.
We press first.
Yeah.
And then the valedictory will do, है ना? Because somebody has to go.
Sure.
Okay. Because your train is running 4 hours 30 minutes.
30. चलिए.
चलो ये okay. Thank you sir.
Yeah, okay. चलिए आइए.
ये आप ये पैकेट वाला बांटिए इसका और ये चाय देना शुरू कर दीजिए.
चाय शुरू कीजिए.